Hey team, Dr. Jack Orty, and in the previous videos I've been building up to this idea that neuroinflammation is a major driver of Alzheimer's disease pathology. It's one of the fundamental causes of Alzheimer's disease. And in the next couple of videos, in this video and in the next couple of videos, I really hope to drive home some of that key research that links the inflammatory nature of microglia, the immune cells of the brain, to neuronal death and neurodegeneration in Alzheimer's disease. So in this title slide here, I've just got an image of positron emission topography of patients with either normal cognition, mild cognitive impairment, MCI, mild cognitive impairment, or Alzheimer's disease, full-blown Alzheimer's disease. And positron emission topography, or PET imaging, allows you to inject a radio-labeled ligand. Now, a ligand is just a chemical that will bind to something, often a receptor. That's a ligand. And so we can inject these patients with a ligand that we know binds to activated microglia. Now, this ligand is called TSPO, and we radio-label it and we inject it into a patient, and this radio-labeled ligand will bind only to activated microglia. So now we can view live in patients how active the microglia are. So here we can see it's sort of a heat map, so the more red, the more active they are. In a normal cognitive functioning adult, we don't see a lot of activated microglia. In mild cognitive impairment, we see some activation of the microglia, and in Alzheimer's disease, we see a lot of activation of the microglia. Now, studies have actually shown that the level of activation in the early stages of mild cognitive impairment actually predicts how likely you are to go from mild cognitive impairment down to Alzheimer's disease, and how steep your cognitive decline will be during Alzheimer's disease. So that's a really strong indicator that there is a causative link between neuroinflammation and Alzheimer's disease. But I'm a cellular biologist, so let's start to jump into some of the molecular and cellular interactions that are leading neuroinflammation to cause neurodegeneration and Alzheimer's disease. So let's jump into it. So this is roughly the disease model of Alzheimer's disease that neuroinflammatory scientists are working with. And it's a modification of a very famous model called a Jax model. That's not named after me, it's named after someone with the last name of Jax, who published this sort of model, demonstrating that this is seems to be the pathology progression of Alzheimer's disease. So the first thing we get in Alzheimer's disease is amyloid growth. And this seems to happen early on, much before cognitive decline. So if I was to put some estimates on the time here, I would say this is happening when you're 50 years old, you get amyloid buildup. Now, after amyloid buildup, we get inflammation and neuroinflammation, and we know from several experiments, neuronal inflammation is sufficient to induce tau pathology. So after the inflammation, we will get tau pathology. Now, this would happen even if the amyloid wasn't there and we had neuroinflammation for other reasons, but that wouldn't be Alzheimer's disease. So if you had, for example, chronic traumatic injury of the brain, which is called CTE, chronic traumatic encephalopathy, we would have inflammation leading to tau pathology, and we wouldn't need this amyloid part of it. So in Alzheimer's disease, we get this amyloid growth, then we get inflammation, then we get tau pathology, that's phospho-tau and tau tangles, and then we get neuronal dysfunction. Neurons can't function very well with tau tangles inside their axons. They can't get transport down their axons, they lose their connections, they lose their structures, and then they begin to die. And so we actually get neuronal loss as well. And then we get cognitive decline. Your brain can actually handle losing a lot of neurons without obvious cognitive decline. We call this cognitive reserve. It's amazing how atrophied, how much tissue loss we can get in the brain before we notice cognitive deficits. And this is called cognitive reserve. And so essentially we get a loss of, if this is the threshold of neuronal loss that leads to cognitive decline, we have this many neurons. So we get neuronal death, neuronal death, neuronal death without really noticing much changing. And then once we get to here, we start to get cognitive deficits. And as more and more neurons die, we start to get cognitive deficits. And this gap here is called cognitive reserve. We can handle quite a bit of neuronal loss, and then we start to get massive cognitive decline. So this is called the Jax model. And what we're going to be talking about here is this inflammation. How does the inflammation lead to tau pathology and neuronal dysfunction? All right, so just a reminder, these are your microglia, and these are healthy microglia, and they're out there surveying the brain with their long processes. And here we have some tissue damage, and this is what microglia do when there's some sort of damage or pathogen or some sort of molecular signal of inflammation. They surround the damage, and they actually lose their processes. They bring their processes in, and they become active and inflammatory. And microglia can do a lot of things. First of all, they can release toxic effectors, they can release reactive oxygen species, and that's them trying to sterilize the environment. If we have an infection going on, microglia can do things like release peroxides or hydrogen peroxide, for example, to sterilize the area. But microglia can also directly eat neurons, and this is what we're seeing here. In green, we've got a neuron. In red over here, we have a microglia, and this is on day 0, and this has been viewed in a mouse with a cranial window and a special 2-photon microscope looking down at the fluorescently labeled neurons and microglia. So this is a live mouse being viewed over several days. We can see here the microglia is coming over, and eventually it ends up on top of the neuron. It's not the crispest images, but you have to remember this is going through the skull, through a little hole in the skull with some clear gel down into the brain. And now we can see the microglia is on the neuron, and then by day 6, the neuron's gone. And that's because the microglia has actually eaten the neuron. Now, we know a little bit how this might be happening. So we have an enzyme that flips a phosphatidylserine, or PS, it's a phospholipid, down into the inner leaf of the membrane. So normally you'll have most of your phosphatidylserine on the inside and none on the outside. But when flippase stops working for some reason, or phosphatidylserine ends up on the outside, we end up with complement 1 protein binding to it, and then that triggers complement 3, which then triggers the binding of a microglia to eat it. So we can deduce that that neuron that we saw getting eaten by a microglia had its phosphatidylserine flip out. And that can happen for a lot of reasons, but one of the major ones is a reduction in ATP. In order to maintain phosphatidylserine on the inner leaf with no phosphatidylserine on the outer leaf, we need ATP to move those phospholipids onto the inner leaf, and that requires ATP and the enzyme flippase. So if you run out of ATP, the phosphatidylserines will flip out, and now we can get complement 1, complement 3, and a microglia coming in and eating that neuron. But this is actually all healthy, right? So here it's eating a dendrite. Microglia have got to do this as part of the healthy brain. They've got to prune unnecessary synapses. They've got to clean up neurons that are disconnected and dysfunctional, because there's a certain amount of breakdown of neurons throughout your life, and they need to be cleaned up safely by microglia. So this is actually a normal, healthy process when it's at appropriate levels. But I'm going to take you through some experiments to show that amyloid can put this process into hyperdrive, and we end up with lots of synapses and lots of neurons getting fully eaten up by the microglia in a way that is beyond what's normal, and it ends up with a massive amount of neuronal death. Now, to do this, what they do is they did primary cell culture. Now remember, primary cell culture is cells taken from a living animal, rather than immortal cell culture, in which the cells are cancerous. They came from a living animal, but now they will propagate for decades after they've been removed from the animal. So primary cell culture we kind of do within a couple of weeks after taking those cells from a living animal. Now what we actually do is we get a mouse or a rat, and we take their pups. This is a sensitive subject, I get it. We take their newborn pups and we humanely anesthetize them and kill them so they don't experience any pain. And we take their brains out and we essentially mush the brains up. There's no good word for it, I'm sorry. We mush their brains up and we put that into growth media. Now, the reason why we use pups is in adult mice, the neurons are so set in stone, they're so non-proliferative, they're so myelinated, they're so built into the structure of the brain and not plastic, they become less plastic in adult mice, that if you tried to do this, the neurons wouldn't survive the process. But in a young mice, neurons can survive this process. So in a newborn mice, because the brain is so plastic at that point in time, it can handle being turned disassociated into single cells and plated out. So we put a little enzyme in there, collagenase, which digests all the collagen to break down the extracellular matrix so the cells can flow out into the medium. But now we have a mixed culture of astrocytes, microglia, and neurons all growing in dishes or plates. Here we can see there's a cell culture plate with growth media on it. And we're incubating those cells. And now we can perform experiments and view what's going on very precisely, very accurately. We can alter things in the media. We have complete control of the experiment to understand what's going on as we manipulate different things in this experiment. So there's a very cool way to do experiments. So for example, you could alter it, perhaps put amyloid in one well and no amyloid in another well, and then you could look at the neurons. You could count the synapses on the neurons, or you can measure the electrical activity of the neurons. Another thing you can do is look at the phagocytotic properties of the microglia. Are the microglia eating the neurons? And you can look at that in a dish live and watch what's happening. So it's a very powerful technique, and that's what they did in this paper. And then these are some of the results. So this is, remember, a mixed culture of neurons, astrocytes, and microglia. And over here we have the number of neurons per field of view, and it's percentage control. So obviously we get 100% in the control group. It's 100% control. Now in the dark bar we have alive, then we have apoptotic and necrotic. So this was just, they attempted using stains to quantify are there any neurons currently undergoing apoptosis. Now, but more importantly is the height of the bar. How many neurons are actually left in the dish? That's the more important question. So here we can have the control group. Here they put the amyloid monomer in, and they see a massive reduction of neuronal number. So you can see we've got 30% neuronal death just by putting the amyloid monomer on. And this is in the presence of microglia and astrocytes and neurons. Now, an important point here is the monomer will oligomerize very quickly. So even in the course of 24 hours, you're going to get a lot of oligomers forming as well. So even though it says amyloid monomer, it will be a mixture of monomer and oligomers because it'll oligomerize during the experiment. And here they intentionally put on oligomers. Now to do this, they take the amyloid monomer, they put it in some fluid, and they let it oligomerize for a bit before they then apply it to the cells. And over here, they've got fibrillar amyloid. So for that, you incubate it for a very, very long time until it precipitates, and you end up with those fibrils that are not floating around in the media anymore. And you can take those fibrils and put them on the cells. And here we can see we get neuronal death under all of those conditions. So amyloid is toxic in mixed cultures, neurons, astrocytes, and microglia. Okay. So next they wanted to look at dose, and so they used a very low dose. People would probably consider this a low dose, nanomoles. It's a bit controversial. Some people think picomoles, which is even smaller, is probably a more appropriate concentration. It's a little hard to know because directly around a plaque we get a concentration gradient of amyloid. So it's a little hard to know about what's physiologically relevant. But this is a very large dose, and this is I'm going to call this a medium dose. They called it a low dose. I think that's a medium dose. That's a large dose. I think picomoles is probably more of a low dose. Okay, so here they have their medium dose and their high dose, and they show that, yes, we've got less neurons. So again, on the y-axis, we've got neuronal number. We get that 30 to 40% death rate in the presence of amyloid. However, they then, before they seeded out the cells, they took out all the microglia. So now when they're plating out the cells, there's pretty much only neurons and astrocytes. There's no microglia in the flask there. And now they repeated the same experiment with amyloid. And what we see is that this medium dose of amyloid is now not toxic at all. So if you remove the microglia, the amyloid is now not very toxic. We don't get that neuronal loss in these cell culture dishes. However, they did show that the very high dose, the 10 micromolar dose of amyloid, was still neuronal toxic. So it seems like a very, very high dose of amyloid is neuronal toxic, but a medium to low dose is not directly neuronal toxic. You needed the microglia there for it to be neurotoxic. Very interesting result there. Very cool. And now, so I mentioned this, right? The flippase. The flippase is involved in putting the phosphatidylserine in. So if you want to know how are the microglia killing the neurons, one thing you could do is look to see if the flippase has been altered and to see if we're getting that phosphatidylserine flip on the neurons, right? So if the neurons have got the phosphatidylserine flip, perhaps their flippase isn't working, then they'll now get the complement 1 binding protein binding to it, and then they'll get the microglia phagocytosing the neurons, and that might explain why there are fewer neurons in the dish. Perhaps amyloid is inducing the phosphatidylserine flip, and then that's leading to microglial phagocytosing the neurons, and that's killing the neurons, so now we have less neurons, okay? That's what they want to test. Now, there is a family of proteins called annexins that bind to that phosphatidylserine, and that blocks the complement 1 protein binding to it. So if you put annexin into the flask, it will bind to all the PS, the phosphatidylserines that have flipped out, and it will block the complement from binding to it, which will block the microglia eating the neurons. So if annexin protects the neurons from being eaten and dying, we now know that the primary way the neurons are dying is being phagocytosed in a complement-dependent way by microglia. Okay, that's the experiment. Here we go. So this is the experiment. So here we've got the amyloid, number of neurons, control, amyloid, killing a whole bunch of neurons. Again, 30 to 40% of neurons are dying. Remember, this is in the presence of microglia. And here they're put on annexins. So they're put on the annexin protein to block the microglial phagocytosis of the neurons that's phosphatidylserine dependent. And what we see is annexin completely blocks the number of neurons dying. It's returning back to 100%. So now we know that the annexin flip is, the phosphatidylserine flip is involved in the neurons dying due to microglia. Okay, so another thing we can do with annexins, that protein that binds to phosphatidylserine, is we can put a fluorophore on it. So we can put something that will fluoresce green fluorescent protein, for example, GFP. We can put that on the protein and now see are the neurons lighting up green. Now, a little important twist there is that only if you get the phosphatidylserine flip can the annexin bind to it. The annexin can't go into the cell and bind to it from the undersurface. So we can actually make sure that it's only an external phosphatidylserine because proteins can't get into the cell. And again, what we're proposing here is that in a normal situation, there is no phosphatidylserine on the outside, so there should be no annexin binding and no green fluorescence going on. So here we have the amyloid and here we have the control. So we're still getting some neuronal death. Remember, the brains were mushed up and plated out. We do have some phosphatidylserine facing outwards, but when we add amyloid, it goes up by an order of magnitude. You know, there's tenfold brightness over here. So we can see lots of fluorescent annexin binding. So we know that amyloid is inducing a phosphatidylserine flip, and that phosphatidylserine flip is essential for neuronal death. So we now know that the microglia are grabbing onto the neurons that have a phosphatidylserine flip and are eating them. And that's what's causing a 30 to 40% reduction of neurons very quickly. So how is this happening? Well, this paper didn't go into it, but we know a wee bit about how this might be potentially happening. So first of all, this is a bit tough. It does depend on concentrations. We're not 100% sure, but there does seem to be a direct effect of amyloid on the neurons that might be resulting in a phosphatidylserine flip. They seem to stress the neuron out, possibly by permeabilizing the membrane, so making the membrane a bit porous. And this leads to a bunch of things. If you make the membrane a little bit porous, you get reactive oxygen species being produced, you get mitochondrial dysfunction. Those two things are highly linked. When a mitochondria becomes dysfunctional, they release reactive oxygen species. You can think of mitochondria as using reactive oxygen species to generate ATP. And so it's kind of dangerous. It's a little bit like a nuclear power plant. It's using this very powerful tool to generate electricity. But if it breaks, it's going to cause some problems. And so that's what happens with mitochondria. They're trying to use reactive oxygen species to create ATP. they have electrons flying around like mad, and so when you make a dysfunctional mitochondria, it often releases those reactive oxygen species, and you also get loss of ionic control in the membrane, and that's very stressful for a neuron to not be able to control the sodium, and stressful for any cell to not be able to control the ion flow and thus the water flow. Remember, if an ion flows in, so does water. That can actually cause the cell to swell. So there's a lot of problems with losing control of your membrane. And all of this can cause a breakdown of ATP production, which is going to cause problems, right? You're going to end up with less ATP, less flippase activity, and the phosphatidylserine flip, right? So by making a cell dysfunctional, you're not going to be able to maintain flippase activity, and you're going to get the phosphatidylserine flipping outwards. All right. But there's also this indirect effect. We know that amyloids, this is probably a lot more established than the amyloid directly affecting the neurons effect. Amyloid is definitely inflammatory for microglia. Microglia release reactive oxygen species because they're trying to sterilize the area. And so they can actually, in doing so, releasing reactive oxygen species as well as inflammatory cytokines, which act directly on neurons as well. I covered that in the tau video previously. We can end up with the microglia damaging the neuron, which might then cause the phosphatidylserine flip, which might then trigger the microglia to eat the neuron. So again, we can get microglia releasing reactive oxygen species, which damages the neuron. It loses ionic control of the membrane. So when reactive oxygen species interact with phospholipids, they prevent them from doing their job, which is acting as a membrane. It's called lipid peroxidation. And then we can get mitochondrial dysfunction, and that's going to lead to a phosphatidylserine flip in the neuron, right? So a lot of this is about, if you damage the neuron enough, they may be not been producing ATP, and by not producing ATP, they can't run their flippase anymore, and so they're going to get the phosphatidylserine flip. They're not going to be able to maintain phosphatidylserine concentrations on the inner leaf. They're naturally going to flip out. But there's actually another effect going on, and it's another brilliantly named enzyme. Is all of these things, reactive oxygen species, mitochondrial dysfunction, and loss of membrane control, ionic control in the membrane, activate an enzyme. And they activate an enzyme that mixes up the leaves, the inner and outer leaf of the phospholipid membrane of the bilayer, right? So they actually mix it all up. They scramble the phospholipids, and that's the job of this enzyme. See how it's just scrambling the phospholipids there? And its name is scramblase. Isn't that amazing? So you got flippase, floppase, and scramblase. And that's brilliant. Those are some fantastically named enzymes. So reactive oxygen species, mitochondrial dysfunction, and loss of ionic control, particularly calcium, can activate scramblase. And now the scramblase is going to cause those phosphatidylserines to flip out. And that will allow the complement to come, and then the microglia will come and gobble up the neurons. So that's a lot of what's going on. What we can see from this paper is that amyloid is mostly toxic through microglia and through specifically microglia phagocytosing neurons, possibly by stressing them out first by squirting reactive oxygen species onto the neurons, and then phagocytosing the stressed out neurons. But if you block that phagocytosis, the neurons don't die in this short time frame when being exposed to amyloid. So microglia, yeah, they're surveying the environment, and then they're looking for pathogens, but then they seem to respond to amyloid. And in a later video, very soon, I'm going to cover why do microglia respond to amyloid as if it's a pathogen. They seem to be thinking amyloid is a pathogen and releasing those reactive oxygen species and initiating phagocytosis and all that jazz. So the microglia are surveying the environment, and they detect amyloid, and they kind of think it's a pathogen, and they start responding as if it is a pathogen. They then release toxic effectors, so these are non-specific killing chemicals, such as peroxides, like hydrogen peroxide, trying to kill the supposed bacteria out there, but they end up damaging neurons. They then phagocyte, so they normally would eat bacteria or dying cells, but they've actually induced a bunch of dying cells, and this is all through phosphatidylserine, and then they come eat it. They come eat those phosphatidylserine signaling cells. The cells are telling them, come eat me, because of that phosphatidylserine flip. But another thing microglia do is they're gatekeepers. They invite immune cells, peripheral immune cells, into the brain. So the brain normally is relatively immune privileged, not a lot of immune cells in the brain. Microglia can invite peripheral immune cells into the brain, and that can cause a huge problem. And that's what I'm going to cover in the next video, neutrophils and Alzheimer's disease.